Okay, good morning children. The seventh chapter, third unit, seventh chapter continuation. Sexual reproduction in flowering plants. Subtopic. Now we are going to discuss about the stamen, microsporangium, and pollen gland. Okay, children. Now what is the structure of the stamen? Okay, but what is the structure of the stamen? Here you can see structure of the stamen. This is the structure of the stamen. Structure of the stamen. Okay, now this is the filament. This is the filament. This is the pollen. This is the anther, Nani. Anther. This is the anther. This is the anther. So here you can see clearly each stamen having mainly two parts. One is stalk-like filament. Another one is at the end of the filament you can see anther. Okay. So here filament and anther, Nani. Filament especially you can see long, slender, stalk-like structure, that is the filament, and the filament having bilobed, here having two lobes, Nani. Bilobed anther, bilobed anther, bilobed, here having two lobes now, two lobes, bilobed anther is there at the end of the filament, bilobed anther is there. Okay, so here especially you can see in angiosperms, a typical angiosperm, anther is a bilobed structure, bilobed, two lobes are there, each lobe having two sac-like structures, each lobe having two, inside each lobe having two sac-like structures, two sacs. Okay, means two theca sac-like structures. Okay, they are dithecous. They are dithecous. Two lobes are there, each lobe having two sac-like structures. That's why they are called dithecous. They are called dithecous. 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 Okay, they are dithecous. Okay, and generally you can see a longitudinal groove runs lengthwise. They separate these two lobes. These two lobes are separated in some flowers, especially. These two lobes are separated through a longitudinal groove. A longitudinal groove which runs lengthwise, which can separate the two sacs of these lobes. Okay, especially in some flowers, a longitudinal groove which grows lengthwise and which can separate these two sacs. Okay, so especially generally dithecous, two lobes are there, but especially in hibiscus flowers, the anther of hibiscus is a single lobed, hence it is called monothecous. Bilobed means dithecous. Especially in hibiscus flower, the anther having only one lobe, that is called monothecous. Monothecous. That is called monothecous. That is called monothecous. Best example, hibiscus. Best example, hibiscus. Best example, hibiscus. Best example, hibiscus. So, the bilobed nature of anther is clear. You can see in the transfer section of the anther. While in the transfer section of the anther, you can see the bilobed structure of the anther, each lobe having mainly two sacs. Okay, so the anther is four-sided structure. Okay, tetragonal. The anther is a four-sided structure, tetragonal structure, and this is consisting of four microsporangia located at these centers. At these centers, you can see four microsporangias. Four microsporangias are present in these four sides. Okay, the anther is having four sides, tetragonal. Each side you can see one microsporangia is located. So total four microsporangia are located at four corners. Okay, four microsporangias are located at the corners. Okay, and each lobe having two chambers now, total four chambers. Okay, so these microsporangia develop and become pollen sac. These microsporangia only develop into pollen sac. Okay, four corners are there, four sides are there. These sides having four microsporangia. These microsporangias are developed into pollen sac. Okay. Now structure of the microsporangia. Now what is the structure of the microsporangia? Now the structure of the microsporangia. Now the structure of the microsporangia. But what about the structure of microsporangia? Structure of microsporangia. But what is the structure of the microsporangia? Structure of microsporangia. Here you can see, Nani. Before only I said four sides are there, two lobes are there. Each lobe having two sac-like structures. No, that is called microsporangia. Here you can see clearly transverse section of a young anther, TS of young anther. You can see here you can see the TS of anther. The TS of anther having the TS of anther, you can see the TS of anther is having mainly okay, four sides, Nani. Each side having okay, four sides are there. Okay, four sides are there. So here you can see, Nani, four sides. Okay, so especially you can see this is a connective. In the middle you can see microsporangia. Okay, sporogenous tissue is there. Sporogenous tissue is there, sporogenous tissue is there, and epidermis. Epidermis is there, endothelium, okay, epidermis, endothelium, middle layer. This is the middle layer. This is the tapetum. Okay, so this is about the TS of anther. TS of anther. TS of anther. Okay, so the our wall layers, other diagram also there. And what about the structure of the wall layers? Structure of wall layers diagram also important. But here you can see epidermis, endothelium, middle layers, tapetum, microspore, mother cells. Okay, here somewhat layers are there on here. Somewhat layers are there. Okay, this is the epidermis, endothelium, and middle layers are there. Middle layers are there. Middle layers are there. Okay, middle layers, especially you can see middle layers are there. Then tapetum and microspore mother cells also there. Okay, middle layers. Middle layer, tapetum, and this is the microspore mother cell. Microspore mother cell. Okay, so these are the layers. So especially you can see microspore mother cell. So the structure of the microsporangium. But what about the structure of the microsporangium? You can see the transverse section of a typical microsporangium appears near circular structure. Here you can see the circular structure of the microsporangium. So it is generally surrounded by a four-wall layer. The microsporangium, which is TS, transverse section of microsporangium, which is appear like a circular structure, which is surrounded with four-wall layer. But what are the four layers, four walls? First one is epidermis, second one is endothelium, third one is middle layer, fourth one is tapetum. These are the four layers. Endothelium, epidermis, endothelium, middle layers, and tapetum. So here, these first three layers, epidermis, endothelium and middle layer. These first three layers, they can perform the function of protection, they can give protection, and also they help in dehiscence of anther, breakup of anther. The first three layers they give protection and also they help in dehiscence, means splitting, dehiscence of anther to release the pollen, to release the pollen grains. Three layers, first three layers they can give protection and also they are very useful to help in dehiscence, breakup of the dehiscence of these layers, and they can release the pollen grain. Okay, dehiscence of the anther, then they are helpful in releasing of pollen grains. So the epidermis is the one-celled thick, the cells present between the pollen sac, the cells which are present between the pollen sac, the cells which are present between the pollen sac, the cells which are present between the pollen sac that are thin walled. Okay, the region is called stomium. The region is called stomium. The region is called stomium. Here the stomium is there. Stomium. The cells which are present between the pollen sac. The cells which are present between the pollen sac, they are very thin walls. The region is called stomium, which is the stomium is very useful for the dehiscence of the pollen sac. On the stomium only, the pollen sac will split and releasing of the pollen grains. And one more thing here you can remind, epidermis is one cell thick, epidermis is one cell thick. Okay, the cells which are present between the pollen sac, the cells are which are very thin, and this region is called stomium. On the stomium only, the pollen sac dehiscence and release the pollen grains. Okay, so the stomium is very helpful for dehiscence, means splitting of the pollen sac. Okay, so this is about the epidermis and stomium. Epidermis is one cell thick, the cells between the pollen sac that are called, which are very thin, that are called stomium. The stomium is very useful for dehiscence of pollen sac and releasing of anthers, releasing of pollen grains. Okay, next here epidermis after stomium, after epidermis after you can see endothelium. The endothelium is below the epidermis. It expands, which expands radially, which is having fibrous thickness. Endothelium is present below the epidermis and which expands radially with fibrous thickness. So after maturity of these cells, they can lose water, they can contract, and they help in dehiscence of pollen sac, anther, they can dehiscence. So especially endothelium, after maturity of these endothelium cells, they can lose the water, they can contract, and they helpful in dehiscence of pollen sac, anther. Okay, and one more thing here you can remind, the endothelium expands radially, which is having fibrous thickness, which expands radially with fibrous thickness. Okay, this is about the epidermis, stomium and endothelium. Next, but what about the tapetum? Tapetum is the innermost layer, the inner layer is called tapetum. The innermost layer is called tapetum. It is very useful for, it provides nourishment, nutrients. It nourishes the development of the pollen grains. While developing of the pollen grains, they can get nutrients from the tapetum only. Tapetum which nourishes the development of the pollen grains. While developing of the pollen grains, they can get nutrients from these tapetum only, which nourishes the development of the pollen grains. That is the innermost layer, that is the tapetum. Okay. The cells of the tapetum cell contain dense cytoplasm and they have more than one nucleus. They have dense cytoplasm. This is the innermost layer tapetum. Here, when the anther is young, in that young anther, some cells, the group of compactly arranged homogeneous cells, same cells. Whenever the anther is young, a group of compactly arranged cells, homogeneous cells that are called sporogenous tissue, which occupies at the center of microsporangium. At the center of the microsporangium, you can see sporogenous tissue. At the center of microsporangium. You can see sporogenous tissue. How these sporogenous tissues are developed? These are compactly arranged homogeneous cells, means same cells. Homogeneous cells are compactly arranged, they form sporogenous tissue, which occupies at the middle of the microsporangium. Okay. So these are the explanation of the four type of the layers. This is also the location of the four walls: epidermis, endothelium, middle layer, tapetum, and microspore mother cells. Okay, microspore mother cells. Next topic, Nani, continuation. Microsporogenesis. Next topic, continuation. Microsporogenesis. Next topic, continuation. Microsporogenesis. But what is microsporogenesis? Okay. As anther develops the cells of the sporogenous tissue, while the anther develops the cells of the sporogenous tissue, the cells of the sporogenous tissue, while the anther develops the cells of the sporogenous tissue undergo meiotic division. The cells of the sporogenous tissue undergo meiotic division. The cells of the sporogenous tissue undergo meiotic division. They form microspore tetrads. They form microspore tetrads. Means four microspores are there. They form microspore tetrads. So this process is known as microsporogenesis. Okay, by the development of the anther, the sporogenous tissue undergo meiotic division and they can produce microspore tetrads. This process is called microsporogenesis. Okay. So here each cell of the sporogenous tissue is capable of giving rise to microspore tetrads. Each cell of the sporogenous tissue that able to produce microspore tetrads, so that each cell of the sporogenous tissue able to produce microspore tetrads. So that cell is called primary microspore mother cells. Pollen microscope mother cells. So each cell of the sporogenous tissue able to produce microspore tetrads. So the each cell we call microspore pollen or microscope mother cell. Pollen or microscope mother cell. Okay, microscope mother cell. So the process of formation of microspores from the pollen mother cell, that is called microsporogenesis. Okay, the process of formation of microspores from the pollen mother cell through the meiosis. Through the meiosis division only. Through the meiosis division only. The process of formation, the process of through the meiosis division only. Through the meiosis division only, the process of formation of microspores from pollen or microspore mother cells. The process of formation of microspores from microspore mother cell through the meiosis, that is called microsporogenesis. That is called microsporogenesis. Okay, that is microsporogenesis. Next, what about the structure of the pollen grain? If you see the structure of the pollen grain, but what about the structure of the pollen grain? Now structure of the pollen grain, microsporogenesis after structure of the pollen grains. Okay, Mari, what about the structure of the pollen grain? Structure of pollen grain. Structure of pollen grain. So if you see the pollen grain represent the male gametophytes. Pollen grain represent the male gametophyte. Pollen grain represent the male gametophyte. If you touch the opened anthers of hibiscus, whenever if you touch the opened anthers of parent sac, opened anther of hibiscus, okay, or any flower, you would find a deposition of yellowish powdery pollen grains on your fingers. If you touch the open end of the pollen anther, you feel deposition of yellow powdery pollen grains on your fingers. So if you see through, okay, so sprinkle these grains in a drop of water on the glass slide. You can observe it under the microscope. You will see different varieties, different size of the shape, different size, shape and color of the pollen grains. Okay, so pollen grains are generally spherical structure, which measures about nearly 20 to 25 micrometers in diameter. Diameter is 20 to 25 micrometer diameter, which is spherical structure. Okay, so it has a prominent two-layered wall. Each pollen grain having two-layered wall. The outer wall, which is very hard, that is called exine. The outer wall is called exine. The outer wall is called exine. But what about the inner wall? The inner wall is called intine, exine and intine. Outer wall is very hard, which is called exine. The outer wall of the pollen grain, which is very hard, that is called exine. Inner wall is intine. But what about the exine? The exine is made up of sporopollenin. The exine is made up of sporopollenin. The exine is made up of sporopollenin. Exine is made up of sporopollenin. Okay, which is one of the most resistant organic material. Which is one of the most resistant towards the organic material, which is one of the most resistant organic material, and it withstand high temperature. It can able to survive, it can withstand high temperature and strong acid and strong alkalines also. Okay, which is one of the most resistant organic material and which can withstand high temperature as well as high strong acids, high alkaline conditions also it can withstand. So pollen grains exine are the prominent apparatus. They are gene pores. The pollen grain exine has a prominent apertures. The exine having some apertures, gaps are there. These apertures, the exine outer layer is exine. The exine having some apertures, gaps are there. These are called germospores. These are called germopores. These are called germopores. The exine having some aperture, these are called germopore, where sporopollen absent. Already you know that the outer layer is called exine. The exine is made up of sporopollen. Because of this sporopollen, it can withstand in high temperature, strong acid, which can resist into organic acids. But here, the pollen grain, the outer layer, in some areas, the outer layer having apertures, gaps, that are called germopore. In that germopore, there is no sporopollen. On the outer layer of the pollen grain, the regions where the sporopollen was absent, the regions appear like a small apertures, gaps, that are called germopores. Okay, these are called germopores. Pollen grains are well preserved as fossils because of the presence of sporopollen. These pollen grains are very preserved as fossils because of the presence of sporopollen. So this is about the external layer, which is very hard exine. Now what is the inner layer? What is the innermost layer? Intine. Exine and intine. So about the inner layer we are going to discuss in tomorrow class. Okay, thank you children.